Amyotrophic lateral sclerosis, or ALS, is a progressive, fatal degeneration of both upper and lower motor neurons. Upper motor neurons transmit information within the central nervous system, while lower motor neurons do so within the peripheral nervous system. In a neuron, a signal is received by the branch-like structure of the dendrites. This signal then passes through the soma and travels down the axon. Fatty myelin sheaths act as an insulator to ensure that this signal is not lost. Along the axon's membrane are voltage-gated channels such as the sodium-ion channel. When the voltage-gated channels receive a stimulus strong enough to overcome the thresholds, the gates open, allowing positively charged sodium ions to rush into the negatively charged cell, depolarizing it. As more and more gates open, the polarity of the cell will reverse, with the inside being positive and the outside being negative. Potassium channels will then open to allow the membrane potential to return to its original state. This action potential propels the nerve impulse throughout the neuron. The upper motor neurons then transmit the nerve impulses via the neurotransmitter glutamate, which is received by the lower motor neurons' glutamatergic receptors. The upper motor neuron is the presynaptic cell and the lower motor neuron is the target cell or the postsynaptic cell. The presynaptic end contains vesicles filled with glutamate. When the nerve impulse reaches the axon terminal, calcium ions enter the membrane of the presynaptic end, associating with proteins in the outside surface of the vesicles. This allows the vesicles to exit the cell via exocytosis and enter the synaptic cleft. Glutamate crosses the cleft, binding to its receptors, creating an electrical change across the membrane, which is then carried throughout the cell. At a neuromuscular junction, the lower motor neuron makes contact with the muscle, relaying the signal to contract. Patients with ALS are unable to send these signals to their muscles due to the degeneration of their neurons. Because they are no longer being used, the muscles atrophy and eventually become paralyzed, and respiratory failure eventually leads to death. Scientists have not been able to pinpoint the exact causes of motor neuron degeneration, but they have some ideas. In a healthy neuron, glutamate will be disposed of once it has passed the postsynaptic membrane. However, prolonged excitation of neurons can lead to a buildup of glutamate, which can be toxic to cells. In addition, patients with ALS seem to have changes in their mitochondria, which could be an indicator that mitochondrial damage could be a factor. Lastly, misfolded SOD1 proteins may build up in the cell, impeding cell function. While there is no cure for ALS, researchers are hopeful that interrupting apoptosis or programmed cell death may stop the death of neurons. Treatments such as riluzole or edaravone have shown to slow the progression of ALS. Riluzole works by decreasing the amount of glutamate in one's body, while edaravone protects neurons from toxic substances called free radicals. Hopefully in the future we can find a cure for this disease. Every 90 minutes someone is diagnosed with or dies from ALS. To help support the road to a cure, please consider donating to the ALS Association.